Welcome to Dentistionary. Hope so you guys are now getting used to this field, and I'm sure you will soon be loving dentistry as well. So, this is our fourth lecture, and our topic for today is clotting mechanism, which is an important long note question for your profs. And this is a very short topic as well. Guys, please post your comments regarding your feedbacks for this channel. These comments are really helpful for us. You all can also ask your doubts in the comment section. Okay, so let's begin our today's topic. First of all, what is hemostasis? Okay, wait. It's hemostasis, not homeostasis. Don't get confused between hemostasis and homeostasis. Okay, let me clear this out. Hemostasis means stopping of bleeding from any wound, which is often the first stage of wound healing. Whereas homeostasis is the body's state of balance or its tendency to maintain a constant and stable state. Getting back to a proper definition of hemostasis, it is the spontaneous arrest or prevention of bleeding by physiological process. Next comes the mechanism of hemostasis. First, the injury to the vessel wall, that means the injury to the blood vessels, which leads to the formation of clots. Then the damaged blood vessels get sealed, and finally there is a prevention to the blood loss. In this way, the clottings are formed. Next, there are three main major events which get involved during hemostasis. They are: number 1, the constriction of injured blood vessels. Next, the formation of a temporary hemostatic plug of platelets. And number 3, the conversion of temporary hemostatic plug into the definitive hemostatic clot. Okay, there's a reminder. We also provide you PPTs of these slides, which is present in the description box. Next, this diagram is very important, which you can include in your answer. Let's give a good impression to the teachers. After the series of events, let's get back to the blood clotting factors. We have already read these factors in our previous classes. There are 13 major factors, namely fibrinogen, prothrombin, thromboplastin, calcium ion, proaxillerin, axillerin, proconvertin, antihemophilic factor A, Christmas power factor or antihemophilic factor B, Stewart power factor, antihemophilic factor C, Hageman power factor, and finally the fibrin stabilizing factor. So it's good if you learn all these 13 factors in a sequence. After this, the most important part, that is the mechanism of clotting. Firstly, the intrinsic pathway and the extrinsic pathway. What are these? Let me tell you. Extrinsic pathway is the shorter pathway of secondary hemostasis, whereas the intrinsic pathway is the longer pathway. Hope so it's clear. Next, in extrinsic pathway, once the damage to the vessel is done, the endothelial cells release the tissue factors, which goes on to activate the inactive clotting factors. Like here you can see, the inactive clotting factor 12 is being changed into the active form 12A. A refers to the active form. Next, clotting factor 11 is converted into the active form 11A. 9 is converted to 9A. Similarly, 10 is converted to 10A. So, we can say it keeps on activating the inactive clotting factors. Whereas intrinsic pathway is activated through exposed endothelial collagen. So these are the main basic differences between the extrinsic pathway and the intrinsic pathway. Rest you can see in this mechanism. It's very clear. If you guys don't understand anything, kindly comment us in our comment box. We'll make sure We will repeat this in our next lecture. Next, we come to the applied, that is thrombosis. Thrombosis, it's a very familiar term. What is thrombosis? Formation of clots inside the unbroken blood vessels, usually a vein, is called thrombosis. Means inside the blood vessels when the clots are being formed, that is called thrombosis. And that clot is known as thrombus. What are the main causes of thrombosis? They are sluggish blood flow, damage to tunica intima by atherosclerotic plaques, mutation in the gene for factor 5. What is factor 5? It's the labile factor or proaxylidine. That's why I said you must learn the factors in sequence. So the mutation in the gene for factor 5, antithrombin 3 and protein S. What else happen in thrombosis? Thrombi or emboli frequently occlude the blood supply to the organ in which they form and damage it. Next, we have the anticoagulants. What are anticoagulants? They prevent clotting. Simply, in simple terms, they prevent clotting. There are two main types of anticoagulants, natural and artificial. First comes to the natural. First example of natural anticoagulant is heparin. What is heparin? It is a powerful anticoagulant that is first isolated from liver and thus it is named so. It is also present in many other organs like lungs. What are the main actions of heparin? It facilitates the action of antithrombin 3, thereby inhibiting the active forms of clotting factors 9, 10, 11 and 12. It is also responsible for anticoagulation property. What are the main origin of heparin? From where it has been secreted? It is secreted mainly by the granules of the circulating basophils and the granules of the mast cells. Heparin is mainly destroyed by an enzyme called heparinase which is present in the liver. There are two main uses of heparin. They are: number one, it is responsible for the fluidity of the blood. The fluidity that the blood gets is because of heparin. And number two, the post operation prevents spread of intravascular thrombosis. Next, we have a short mechanism regarding heparin, which you can see. So, that's it for today, guys. Hope you all enjoyed today's lecture. We'll soon meet you with our next lecture, which will be on the blood group. Till then, keep sharing this video and share lots of support for us. Thank you.